I recently came across a video from a pastor at Village Church, which is outside of Richmond, Virginia. This video was done by Dr. Chris Bolt, who's a pastor of theology and apologetics there at the Village Church. And so he's got this series of videos where he does apologetics and he touches on science and including evolution. So he did a video recently trying to debunk the concept that humans and chimpanzees share a common ancestor, focusing on a bunch of creationist nonsense, specifically around the comparisons of the chimpanzee genome to the human genome. It's filled with a bunch of misconceptions. I'm actually going to be tearing apart this video over the next several videos of mine. There are three major topics that I'm going to be covering. One is his discussion about science and scientism and how we know about the natural world. Two, he also discussed a whole bunch of stuff about endogenous retroviruses. We'll be talking about that as well. But in this video, what we're going to be doing is covering over his concept of common descent versus common design. But before we do that, I want to highlight a major misconception that Bolt has here about the chimpanzee genome versus the human genome, which has been extensively covered elsewhere.
So we want to show or demonstrate or evidence or prove that chimpanzees and human beings came from a common ancestor. But the evolutionary scientists just start with that assumption and then use the evidence in the lab to try to fill that in and then draw these evidences based on it. And so, yeah, if you start with the assumption there's a common ancestor, then you're going to draw these inferences that, in fact, chimps and humans, the genomes there look similar. So you get these numbers like, oh, there's a 98% similarity between chimp and human DNA. Well, even that on a finer level has been called into question. Most people would say 89% now. But there are studies out there that say that the genetics between the chimp and the human are at 85% similarity.
I just can't do it. I can't take this shit no more, man.
Okay, so this is total garbage. 85%, we know that that's not true. This has been thoroughly debunked by Gutsa Gibbon, aka Erica. I've taken a little bit of material from her over 2-hour video on this topic. I encourage you to watch it. She thoroughly breaks down where this creationist nonsense comes from. So let's take a look a little bit of what she says about the similarity between chimpanzees and humans, why it's kind of a complicated topic, but we also certainly know that it's not 85%.
In fact, humans and chimps are more similar genomically than African and Asian elephants are to one another. And not only that, but as more apes were sequenced, it came to be seen that humans aren't just most closely related to chimpanzees, but that chimpanzees are also most closely related to humans. This means that your genome, the individual watching this, has more in common with a random chimpanzee genome than any gorilla on the planet shares with that random chimp. The closest relative to humans and chimps as a pair is then gorillas, followed by orangutans, and then the gibbons. This comparative genomics work is effectively a souped-up version of a paternity test. The genome reveals how close two organisms or two species are, and humans are closest to panins, placing us not just with the other apes, but smack dab in the middle of the family Hominidae. Firmly apes, past the bananas. Creationists went absolutely ballistic at this. And this is because, according to certain, and in my opinion, myopic and incorrect interpretations of Genesis, the sort of Abrahamic God created humans literally in his image out of nothing around 6,000 years ago at the end of a sort of 5-day smorgasbord of creating things from like dirt and literally out of thin air.
Cringe.
But as I'm sure you can understand, it's very difficult to accept human evolution and common descent if you have this particular starting point, as it were. Many, in fact most, Christians and other religious folks make it work. Creationists do not. And so, after the human-chimp genomic similarity number was published around 2005, creationists suited up for several decades of unimaginative whinging. Because if humans are created in the literal image of God, why monkey?
Erica does such a fantastic job breaking down this creationist ridiculousness that the human genome and the chimpanzee's genome is something on the order of around 80% or 85% similar, that I really encourage you to watch the entire thing. It really comes from the work of one young earth creationist, Jeffrey Tompkins. She does original work to show that his conclusions and his methods are completely wrong. Absolutely demolishes this entire idea. Now, the thing is, though, what does it mean to be 89% or 85% or 98% similar? What does that number mean in itself? 98% sounds really, really high, or 99% sounds really, really high, which is, I think, the biggest thing that creationists are most concerned about. But they're missing an important point. 89% similar relative to what? Is 89% high or is 89% low? We have to be able to compare to at least one other organism. As Erica pointed out in her video, that when we compare the human and chimpanzee genomes to each other, we see that they are much more similar to each other than they are to any other ape, be it gorillas or orangutans or gibbons. So we know that therefore humans and chimpanzees had a more recent common ancestor than either of them did with, say, gorillas. That is what the genetic evidence shows. My larger point is that we have to compare multiple species to each other in order to have a sense of what, say, 85% or 89% or 98% difference in genome similarity means. If humans and chimpanzees are 89% similar, okay, maybe that's because of the method that she used, just like what Tompkins did. What Tompkins failed to do was to compare humans and chimpanzees to another species, say gorillas or orangutans. If he had really found that they were 89% similar or 85% similar, he would have found then that humans and chimpanzees would be different from gorillas by a percentage even less than that. And that if you compared all three of these, chimpanzees, humans, and gorillas, to, say, orangutans, then you would have found an even lower similarity score. The similarity score in itself doesn't necessarily mean anything per se. Now, it's important to have very good standards, and Tompkins utterly failed in that process. We really do know that, for instance, the human and chimpanzee genomes is something on the order of around 95 or 96% similar, depending on how you actually count those things. Like, for instance, accounting for indels and many repeated regions, it's around 95% similar. If you're looking just within coding regions, oftentimes you're going to find that chimpanzees and humans are 99% similar or even higher. And of course, that's going to be less so when you compare it with other species, such as gorillas or, say, rodents. So we're going to take a look at an example of comparing primates to the rodents. But first, let's take a look at what Chris Bolt has to say about why the chimpanzee genome is so similar to the human genome. He's trying to really have his cake and eat it too. He's claiming, well, the chimpanzee genome really isn't all that similar to the human genome. But even if it is, there's a reason, which is common design.
Let's say that God exists as we believe, and God has a mind. He is, in fact, a designer, an intelligent designer. Well, this intelligent designer, we believe, would actually have a common plan, as it were, or a common design for those creatures that he actually brings about in the world. And so when we look at, just like with fossil evidence, when we look at genetic evidence and we see these similarities between the two, and we're granting that there's some level of similarity, the natural inference need not be that evolution occurred and that there is common descent. We can say as Christians that, in fact, the similarities are accounted for in that we have a common designer who gave these two different living entities a common design, even though there are also vast differences between the two.
Bolt's argument here is essentially that similarities in genetics is entirely due to a common designer. A designer made human beings, a designer made the rest of the great apes, including chimpanzees. Therefore, they're going to have similar genetics. Superficially, that might actually make some sense. For instance, all the great apes share an enormous amount of morphological characteristics. Superficially, it would seem that common design could be one explanation for why organisms that look very similar would also have similar genetics. And in fact, I can take a clip from Guts of Gibbon's video where she nicely lays out the similarities of the great apes, including humans and chimpanzees.
We humans are apes. We are members of the superfamily Hominoidea and the family Hominidae. This means we are specifically great apes. The reason humans are classified this way is because we have all of the physical characteristics that designate our position with the other apes. Like them, we have internal tails, highly mobile shoulders, a Y5 molar pattern, dexterous hands and wrists, long gestations and long developmental periods, complex social systems, and large brains for our body sizes. We have all the same bones, muscles, and organs, and even our brains have all the same regions, with humans merely emphasizing portions related to cognition. This is why, although all great apes use tools, human tools are the most sophisticated. The difference between humans and other apes is one of degree, not kind. Get it?
Bolt's argument is that similarities in morphology, the fact that the great apes all look very similar to each other, is not due to the fact that they have a recent common ancestor, but that instead they have a common designer. That also is the explanation that he is at least arguing here, that why their genomes are so similar to each other. For instance, a chimpanzee and a human has very similar genomes relative to, say, gorillas or orangutans, but they all are very similar to each other because the designer had to pick from a genetic toolbox in order to make ape-like animals. So of course the genomes are going to be similar. Presumably he would argue the same thing for the other animals, right? For instance, a designer may have made mice and rats or similar organisms, right, from using a genetic toolbox because they needed to look like a rodent-like animal. So the great apes all look similar to each other because the designer decided to use great ape-like genes in order to make great apes. And that's the reason why the genomes are so similar between these two groups of animals and why the genomes are so similar to these two groups of animals. Now, of course, it doesn't fully explain why there's a nested hierarchy, why, for instance, humans and apes are certainly more similar to each other than either of them are to, say, mice or rats, but also that humans and apes are much more similar to rats and mice than any of them are to, say, birds or reptiles or mushrooms. All of life does fit into this nested hierarchy when you look at the genetic evidence. Of course, then he might argue, of course, rats and mice are going to have similar genetics to each other, or similar genetics to the other apes, because they're mammals, and therefore a designer had to use mammal-like genes in order to make the mammals look like the way they look. So the issue is that this argument actually breaks down when you start to look at disparate groups of animals that actually look similar to each other. So I'm going to walk through a series of slides that I made when I had a discussion about this topic with Sal Cordova, a notorious young earth creationist who actually knows a fair amount of, just enough to be dangerous, and fair amount of this, of what genetics is, of what protein folding is. I think that he is completely wrong on just about every conclusion that he draws. But in general, he's a nice guy. He's fun to talk to. And I actually had a conversation with him about this topic that's on my channel. It's not really been seen very much, but you can take a look at it. But I'm going to use the same slides to walk through this argument against what Chris Bolt is talking about. First, we need to talk about potassium channels. Now, potassium channels are a very interesting group of channels that play a big role in the nervous system. Me being a neuroscientist, I know a fair amount about potassium channels. I don't work on potassium channels, but I know a little bit about them. This is an example of one potassium channel. This is the protein structure. This would be indicating the outer membrane and the inner membrane of the cell membrane. So this is a transmembrane spanning protein. This is the protein here. So the protein is, would be rooted into the membrane itself. It's potassium. This is an example of a potassium voltage gated channel from subfamily A, member 1. Okay, keep that in mind. The protein is going to have a pore, so showing that there are potassium ions that can flow through here. That's what those purple dots are. And that there can be when a voltage change happens across the membrane, it's going to change a conformation in this protein, allow potassium to flow through. There are lots of different kinds of potassium channels. So this is the voltage-gated potassium channels. There are SK channels. There are inwardly rectifying potassium channels, the KCSA channels. They all are similar in some ways. They have similar protein structures, but there are also differences in many of them. And then these are big families of potassium channels. They also are similar in that they have a tetrahedral structure, in that there's basically this one protein structure that gets duplicated 4 times in order to make the channel itself, allowing the pore that would either be open or closed, allowing potassium to flow through. Now, potassium channels are very, very important for neurons because they are used to do action potentials. So action potentials allow for the transference of information to be passed along a neuron, eventually getting down to the axonal bouton, allowing for then the synapses to the presynaptic site to release neurotransmitter, which would then act on the next neuron. But the information being passed from the soma, where the dendrites are, and then there's the soma being passed along the axon. The potassium channels are very critical for this process. And so there's this wave of ion flow that happens across the membrane, and potassium channels play a big role in this. And in fact, here's a little diagram illustrating this. So essentially we have 4 steps with ion flow that happens while an action potential is occurring. So initially there's a positive charge on the outside, negative charge on the inside. There is, when a stimulus happens, basically what happens initially is that there's depolarization of the cell, which causes the current to flip. So we allow the sodium ions to flow through the cell membrane into the cell. This results in a positive charge inside the cell. Sodium ions are positively charged. So, right, typically the sodium ions are in high concentration outside the cell, and potassium is relatively high inside the cell. And then, so the sodium ions flow through, so that's where we get depolarization that occurs. If this process happens enough, and there's enough depolarization that occurs, this will trigger an action potential. That leads us to the next stage, where the sodium channels close and then potassium channels open, which allows potassium ions to flow out of the cell, returning the polarization to be positive and then negative. And then we have a sodium-potassium ion channel pump that reverses the flow of sodium and potassium to return it back to this state, which is its resting state. So, and then this is showing the change in voltage that occurs when an action potential occurs. So that potassium channels, the purple ones here, play a big role in this process. So yeah, potassium channels, they mediate these processes. For instance, so this is what, if we were just tracing the change in voltage in a neuron over time, you might increase the membrane potential of a cell. Once it reaches a threshold, that's going to cause action potentials. Those action potentials will spike. It'll go up and down and up and down and up and down until the membrane potential goes back down again. If you use a voltage-gated potassium channel inhibitor, this leads to fewer spikes. You might get some or none, but you can see that in this case with an inhibitor, we basically are decreasing the frequency of action potentials. If you use a potassium channel activator, so something that enhances potassium channels, you actually increase the frequency of action potential firing, illustrating the importance of potassium channels and neuron firing. Now, as I mentioned, there are several different types of potassium channels, right? We got our voltage-gated potassium channels. We got our 2-pore potassium channels, inwardly rectifying potassium channels. And the thing is, these genes have been duplicated over and over again. So there are subfamilies that occur within each one of them. A lot of them are actually not fully well understood. About half of them are well known. We know that the majority of them are found within neurons. So either found in the axon or in the soma, sometimes in dendrites. We know a bit about the various effects of them, but there's a lot that we don't know. So here's a list of all the potassium channels that we don't know. And this is just the voltage-gated potassium channels here, right? So KV refers to voltage-gated, and you can see that it's a long list. And there's a lot of them that we just don't know enough about. Okay, but the thing is, we do know the sequences. That's one thing that we do know. We don't necessarily know how they work. About half of them we do. And we certainly know a lot about the role of potassium channels in regulating brain function. They're very, very well studied. But because it's such a huge gene family, we don't know every single thing. But we can look at the sequence of a given gene or a given protein. In this case, I'm talking about protein similarity, which is essentially going to be very, very similar to gene similarity. So if the gene is very, very similar between, say, chimpanzees and human beings, the protein is likewise going to be very, very similar because genes will code for protein, right? So here I've looked at protein sequences. You can consider this to be reflective of the genetic sequence as well. But if we compare the human protein sequence of KV 2.1 to the chimpanzee protein sequence, one thing that we find is that despite the fact that this protein, I think, has 600 some amino acids, every single one of them is exactly the same between humans and chimpanzees, showing just how similar they are to each other. Now, if we compare the human protein sequence of the KV 2.1 to, say, the house mouse or the rat, well, now we find that there are some differences. There's 93.5% similarity in the protein sequence when we compare the mouse KV 2.1 to human beings. And of course, it's got to be the same if you did it for chimpanzees, since these are identical to each other. So we can always compare one to the other. We can do the same exact thing comparing mouse KV 2.1 to the other species here, right? The mouse KV 2.1 is, of course, going to be 100% identical to itself. When we compare it to the rat, we actually find that they are less similar to each other than, say, chimpanzees are to humans. But what we find is that it's still very, very similar. It's 97.3% similar. The rat KV 2.1 is to the mouse. And if we compare the mouse to the human or the chimp, that they're 93.5% similar. We can do this with the other potassium channels. For instance, if we do KV 6.2, here we finally find some amino acid differences. So, of course, humans compared to itself is 100%. Humans compared to chimpanzees is 99.1% similar. So very, very similar. When we compare the KV 6.2 to the mouse, we find that it's 89.2% similar. And to the rat, it's 90.1% similar. And then if we take the mouse sequence and we compare it to the other species, what we find is that, of course, the mouse is more similar to the rat, 100% relative to 97.3%. And when we compare the mouse KV 2.1 amino acid sequence to humans and chimpanzees, well, they're not nearly as similar. They're only 90.5% similar. As I said, of course, humans have many different potassium channels. And the thing is, we can also compare potassium channels to themselves. And this has allowed us to define different families of potassium channels. So it turns out that, for instance, KV 1.1 family is very, very similar to each other. Then there's the KV 2.1. There's two of those. They're similar to each other. And they're more similar to each other than either of them are to the 3 or the 4. So basically what's happened, what we think has happened, is that these potassium channels have also evolved over time, and through gene duplication, a gene can be duplicated, and then it's going to result in the opportunity for that new gene to evolve and create new roles. I mean, it's still going to be doing very, very similar things, still going to be acting as a potassium channel, but its amino acid sequence can now diverge a little bit and take on new functions in different kinds of cells, for instance, to provide the organism with new specificity. Now, if we look at this, so this is essentially a phylogenetic tree of all the different potassium channels in every vertebrate. And so all the vertebrates have these groups of potassium channels. I imagine there are some subtle differences here and there, that maybe a group of vertebrates has lost one of these subfamilies or something like that. But they're all going to have inwardly rectifying potassium channels. They're all going to have the voltage-gated potassium channels, and so on and so forth. And so what we think has happened a long time ago, long before there was even multicellular life, that an organism, but amongst the prokaryotes or single-celled eukaryotes, well, probably amongst the prokaryotes, which are the bacteria, evolved a potassium channel. And so there was a potassium channel that came about, and then that potassium channel got duplicated, and it got duplicated again and again. And a long, long time ago is when these different groups evolved and diverged from each other within the same organism to create new purposes. And so we can create a phylogenetic tree of the potassium channels themselves. And what this means is that we can compare the human Kv1.1 to other potassium channels within humans. So for instance, the Kv1.1, it has 100% similarity to itself, but when we compare it to the Kv1.2, remember, within the same subfamily, it's 75% similar. But then when we compare the Kv1.1 to, say, the Kv6.1 or the 6.2, well, that similarity goes down quite a bit, right? And then we can also take a different perspective and compare the Kv6.2 to the other ones. Well, Kv6.2 is much more similar than it is to Kv6.1, indicating that it's part of the same subfamily, than Kv6.2 is to either Kv1.1 or 1.2, because that must mean that those subfamilies evolved a long, long time ago, allowing for specialization within the Kv1.1 family versus the Kv6 family, and to give them slightly different functions to provide greater specificity to cells. So every mammal is going to have the same kind of phylogenetic tree for the potassium channels, okay? That means, what leads us to a question: did the designer equip groups of animals to have the same kind of phylogenetic tree? It seems kind of silly that the designer would have to say, like, every single vertebrate would have to have the same phylogenetic tree, indicating that, for instance, all the Kv1.1s in the fish are all very closely similar to each other as they are in mammals. There's no reason a priori why that should be the case. Why, say the mouse or the bird Kv1.1 gene should be more similar to that, like it is in humans, than, say, the Kv1.1 is to the Kv7.1 or 7.4. It doesn't make a lot of sense that all animals should have this same phylogenetic tree if the designer could just kind of give animals whatever potassium ion channels that they need, and why they should all fit within a phylogenetic tree themselves. Okay, so that's just one argument that I'm making here. The evolution model would predict that the genetic code for the potassium channel has been duplicated and then modified many, many times, but there's no reason why an intelligent designer would need to do it this way as well. Why every single organism would have to have this phylogenetic tree of all the different potassium channels within each organism. But how do creationists explain this phenomenon when we comparing the human 6.2 to other organisms, right? So this is getting to the crux of Bolt's argument that he would say, for instance, humans and chimps have a similar potassium ion channel genetic structure and protein structure because of a common designer. That a common designer would design apes to have ape-like potassium channels, and that rodents are going to have rodent-like potassium channels, and that's the reason why, of course, why similar genomes would lead to similar function and overall similar shape and similar morphology. That's what they would argue. So we're going to walk through some examples why this whole argument is completely wrong. And we're going to completely disprove the idea that common genomes, that similarities in genomes must indicate a common designer. This just argument falls on its face. Superficially, this looks like it would be true here, right? Of course, apes are going to have ape genes, and of course, rodents are going to have rodent genes, because the designer is going to design rodents to have rodent genes because they got to have a rodent lifestyle, and then apes are going to have ape genes because they have an ape lifestyle. So let's talk a little bit about rodents, okay? So rodents, this is the phylogenetic tree for lots and lots of different animals. Here are the primates. Humans would be way up here. Here's Rodentia, which includes a lot of different animals, right? So these are the rodents. We got ground squirrels in there. We've got chipmunks. We got beavers, pocket gophers. These are all rodents, but I'm going to focus on a particular group of rodents called the myomorphs, okay? This is what we often refer to as rodents. All of these guys are rodents, as are these. And we're going to focus specifically on these. It includes 6 major groups. There's the jumping mice, the pygmy jerboas, the birch mice, the rats, the true mice, and then the deer mice, okay? Then you can see that they're broken up into 2 sort of groups as well. Now, I've seen lists that the creationists have made of the different kinds, and they often put Myomorpha in a single kind. That's interesting in that must mean that there's, that there was a single common ancestor on Noah's Ark, and that since 4,000 years ago, we've had the evolution of 1,524 different species within just 4,000 years. Just ridiculous on its face. I got a lot to say about that as well. I'm going to make a different video showing why genetics also disproves the idea that you could have common descent over such a short time scale within a given kind, quote unquote kind. But anyway, we're talking about myomorphs, okay, for the most part. And this includes mice, rats, gerbils, hamsters, lemmings, and voles. All of those are in here. So I want to just say up front that rodents look very similar, right? They have a rodent kind of look. They live rodent kinds of lifestyles. There are differences, of course. So here's the Jaculus or the jerboa, one of these weirdo jumping mice, but it's a myomorph. We have, of course, the rat. We've got the mouse. This is a gerbil here. No, I'm sorry, that's a hamster. I think that's a gerbil. So rodents look very similar. They all got these cute little ears, little noses. They are going to look similar to each other. This is true for other groups of animals, right? We can put them in groups. Moles look very similar. They're doing mole-like stuff where they dig in the ground and they all are eating crap underneath the ground. Some of them are interesting and start to stand out. Like this is the star-nosed mole, for which I've actually got a video on my channel talking about interesting neuroscience within star-nosed moles. But, it's still a mole. It's got these big old claws in front of itself and it digs in the ground. So moles all look similar to each other. So we'd expect moles to have similar genetics. Of course, carnivores also look very similar to each other as well. For instance, a fox, a bear, a wolf, a dog, they all have sharp pointy teeth. They all eat meat. They are going to have a particular look as well. And of course, it's not surprising to find out that, for instance, bears, wolves, and dogs have very similar genomes to each other, right? You would expect carnivores to have similar genomes since they all do carnivore-like things. So let's go back to the rodents. All right, rodents, they all look very similar. They have a rodent lifestyle. That would explain why you would think that they would have similar genomes to each other. So if we compare the house mouse voltage-gated potassium channel 1.1 to others. Okay, so right, the house mouse, of course, is going to be 100% similar to itself. Okay, now if we compare the genetics, the protein sequence of the house mouse KV 1.1 to other species, we find the other rodents, we find that it's actually quite high. The KV 1.1 house mouse sequence is very, very similar to the rat, to the hamster, the gerbil, very, very similar across all the rodents. When we compare the house mouse KV 1.1 sequence to humans, we find that it's 98.4% similar. Okay, that would make sense from an evolutionary perspective in that mice and the other rodents would have shared a more recent common ancestor with each other than a mouse did with a human being. Of course, a creationist would argue that, of course, mice and the other rodents should have similar genetics because they are living as rodents and they have a rodent-like lifestyle, and therefore they need rodent-like genes. Now, there's an interesting thing that happens here. When we compare the mouse KV 1.1 sequence to these two guys here, we find that their genetics, their protein sequence and genetic sequences are actually less similar to these two guys than a mouse is to a human being. So humans and mice are actually much more similar to each other on the protein sequence than the mouse is to either of these two species. What do you think could be going on here? I asked the creationists watching, what is your explanation for this? Why would a mouse, which supposedly has rodent-like genes, should be similar to rodents? It's less similar to human beings, but for some reason, it's even less similar to these two. What is it? What is going on here? Let's look at another example. Let's look at the KV 2.1. So the mouse, of course, is 100% similar to itself. When we compare the mouse KV 2.1 sequence to the other rodents, we find that it's less similar than it is to the 1.1, but very highly similar, right? 96%, 97% similar. When we compare the mouse sequence to the human sequence, well, we find that it's less similar than it is to the other rodents, right? It is 93.7% similar. But what happens when we compare the mouse to these two guys over here on the right? 85% similar. Why is a mouse, which is supposedly equipped by the designer to have genetics that would allow it to live a rodent lifestyle, have of course, KV 2.1 channels that are very similar to the other rodents, less similar to humans, but more similar to humans than it is to this guy or this guy, these two species here? What do you think it is about these two species that makes it unique? They look like rodents. They seem to be like rodents. We can do this for all the genes. These are just the potassium ion channels, the voltage-gated potassium channels. You can do it for any gene. I encourage any creationist to go ahead and check my math here. Go to the NCBI database and just compare any single gene. Take the sequence of a mouse gene, compare it to these six species here. You're going to find that the mouse, of course, is going to be more similar to these guys than it is to humans. But you're going to find that the mouse sequence is more similar to humans than it is to this species or this species, despite the fact that this looks essentially just like a mouse. The trick here is that those two species are actually marsupials. Marsupials are a much more diverse group than many people appreciate. Of course, they're not nearly as numerous as the eutherian animals, the mammals, commonly called placenta, which actually is a misnomer because marsupials do develop a tiny little placenta for a very brief period of time. But we do have some of the genomic sequences of some marsupials. So for the Monodelphis domestica, this is an opossum that is found in South America. It's very cute. The thing is, of course, most marsupials do not look very much like rodents. For instance, this is the Tasmanian devil. This is a different kind of opossum. Here, of course, here is the koala. And then this big fuzzy boy is the wombat. They don't look like rodents, okay, right? Of course, they don't. So we would expect them to not have rodent-like genes. So if you take the Monodelphis domestica is the gray short-tailed opossum. It looks just like a mouse. It often acts just like a mouse. It looks basically very rodent-like. Okay. If you compare the short-tailed opossum's KV 1.1 protein sequence to wombats, koalas, the colo opossum, which is the other one that we were talking about, the Tasmanian devil, or the bush-tail opossum, all of these being marsupials, we find that their sequence is around 99% similar to this guy, even though they all look very, very different from each other. But when we compare the protein sequence of the KV 1.1 gene to these guys, all the other rodents, we find that their genetic sequence is less similar to all these rodent-looking guys than these guys. So a wombat, a koala, a Tasmanian devil, a bush-tailed possum, none of them look like this. But their genetics is much more similar to each other than the animals that actually look like this animal. I mean, I could see, you getting this and this confused with each other. This is a house mouse. And yet their genomes are less similar to each other, including even in the protein sequence, the protein coding portions of the genome. They're less similar to each other than they are to the other marsupials. So we can keep going through this. This is the KV 2.1. The short-tailed opossum gene protein sequence, when we compare it to the wombat, the koala, the colo opossum, the Tasmanian devil, and the bush-tailed opossum, we find that it's all, 96% similar. The bush-tailed opossum is 93% similar. It's kind of an outgroup as far as marsupials go. But when we compare the short-tailed opossum 2.1 sequence to the mouse or any of the other rodents. It is less similar than it is to even humans, but also the other marsupials. It is much more similar in its genomic sequence to all these marsupials that don't look anything like this than it is to all these other organisms that do look like this. If you were looking at this, you could argue that you would categorize this as a type of rodent. It lives a rodent-like lifestyle. It is shaped like a rodent. It does rodent-like things. Yet its genetic sequence is less similar to all these other rodent-like animals than it is to any of these other marsupials. So you might argue, okay, you're cheating a little bit. Why don't you're comparing an opossum to a mouse. You're cheating. Okay, a large portion of all the extant marsupials, especially the ones living in South America, of which I think there's something on the order of about 70 species in South America. Most of those are different kinds of opossums. There is something called the yellow-footed marsupial mouse. This looks an awful lot like a mouse. When we compare its NADH dehydrogenase subunit 5 sequence to other marsupials, we find that it's around 80% similar, okay? But when we compare it to the other rodents, it is around 60% similar, less than the other marsupials. This thing is called a mouse. It's a marsupial, of course, but it's called a mouse, and it looks virtually identical to any of these guys. I mean, these guys look like they could be twins. Yet it's only 65% similar to the deer mouse, except when you compare it then to the other marsupials that don't look anything like it, the Tasmanian devil or the wombat. It looks much more similar to those guys than it does to any of the actual rodents. We can do this for all the genes. This is just not restricted to the potassium channels. We could take a different gene. There are families of adrenergic receptors. There's alpha 1, alpha 2, and then the beta adrenergic receptors. We're going to look at the sequence of just one of them. Again, remember moles, they all look very similar to each other. They should all be living mole lifestyles, and of course they do. They dig under the ground, they eat stuff under the ground. What is it about their genetics, though, that makes them unique? So, of course, one of these guys is actually from Australia. This is the southern marsupial mole right here. And its genome has not been fully sequenced as far as I know, but there are some sequences that have been sequenced. So that's why I had to switch over to the alpha-2b adrenergic receptor. When you compare the alpha-2b adrenergic receptor of this marsupial to the other moles, but the placental moles, we find that its genetic sequence is around 70 to 75%. But when we compare it to the marsupials, the wombat, the koala, the short-tailed opossum, the Tasmanian devil, it's around 95% similar. This thing, which looks nothing like these other things, is much more similar genetically to these things than this is to any of the other moles. Again, creationists have to have some sort of explanation for this. If it was really true that there's a common designer that explains why organisms would have similar genetics, because he's using that genetic toolbox to make, say, mole-like animals or rodent-like animals or carnivorous-like animals, then you would expect mole-like animals to have mole-like genes. But no. This marsupial mole actually has much more similar genetically to a wombat, a koala, and a Tasmanian devil than it does to any of the other placental moles. So there's an explanation for this. Evolution provides a great explanation for this. The Australian continent split off from the rest of the continents and became very isolated from the rest of the continents before the dinosaurs died off. Around 130 million years ago, the Australian continent was largely filled with just marsupial animals. There were no placental mammals that were on the Australian continent. It split away through plate tectonics, and it was completely isolated from the other mammal. No other mammal could get to the continent, okay? So what that meant is that the ancestors of all the animals that are living in Australia today could diverge and evolve in similar ways to the placental mammals, right? Australia, it's a big continent. There's, of course, the interior is largely desert, but, over time and, in places along the coast and other things, it's actually quite diverse in the kinds of environments that it has. And so there are going to be available ecological niches for animals to evolve into. For instance, there will always be some sort of role for animals to dig under the ground and eat the stuff that's underneath the ground. Or say eat ants. Ants are ubiquitous across the entire world, including in Australia. So you're going to have marsupial type anteaters that can do this. There is always going to be an opportunity for mammals to live rodent-like lifestyles. Obviously, rodents are extraordinarily successful when you consider how ubiquitous they are across the entire world. It's not surprising that you're going to find that different lineages of mammals, placental versus marsupial, are going to evolve examples of animals that are going to result in rodent-like animals. So the marsupial mouse was almost an inevitability. I mean, there's even flying squirrels versus the flying phalanger, or there's going to be a role for predators, of course, right? So that stem marsupial animal that, got to Australia 140 some million years ago, as the animals diverge and as they occupy various niches, if there are prey animals, well, eventually you're going to have the evolution of some sort of predator, such as the Tasmanian wolf. For instance, we can look at the carnivores, right? So this is a picture of the Tasmanian wolf. It is thought to be at the very least functionally extinct, if not completely extinct at this point. Sometimes there are sightings of the Tasmanian wolf, but no like professional biologist has actually seen them. It's not good evidence that they're still around, but we do have, because they were killed out, I believe, in the 1930s. And so we have their genomes, and that genome has been fully sequenced. There's even a project to try to bring them back, which I think is kind of neat. It'd be awesome if they could, grow them up in the lab and then actually bring the Tasmanian wolf back from extinction. But the thing is, there's actually another marsupial in here as well, the Tasmanian devil. So I tricked you before. Again, these are not all carnivores. These guys are carnivores. These guys are not. But if we were using some sort of genetic toolbox that allows us to say that there's a common designer, and that's why genomes are similar between animals, we would expect, for instance, the protein sequence for the cytochrome B gene from the Tasmanian wolf, that it should be more similar to the other carnivores. But instead, it actually is 90% similar when we compare it to the bush-tailed possum or the Tasmanian devil or the gray short-tailed possum or the marsupial mole. It's 90% similar to those, but it's only 80% similar when we compare them to other carnivores, such as a wolf or a dog or a bear or a fox. Of course, it's going to be less similar to these things because this is a marsupial which evolved separately, and despite the fact that it has entirely the shape, we call it a wolf. It looks like a wolf, but it evolved through the process of convergent evolution, which is why its genetics are more similar to things that actually don't look anything like it than it does to things that actually do like it. So I wanted to show you what Sal's explanation for this pattern was. You can see that it's not a terribly convincing explanation. And in fact, at the end of this debate, you could see the end here, he kind of ran off and had to end it pretty quickly. He said he had a meeting, but yeah, anyway, here's that clip. Okay, so you don't have any explanation for this?
No. Okay. Except a miracle. Only a miracle.
A miracle.
Miracles are not subject to direct.
These animals.
That's the thing. Miracles are not subject to actions. That's why they're.
That's miracles. Your explanation? This isn't science. This is why I started off my debate, because I knew, of course, it's a miracle. I started off my debate that this is not a debate about science. This is a debate about theology, and it really is.
Well, my point is
because it's not about science.
Well, the point is,
we're talking about a lot about science topics, right, but it's about theology. You're going to invoke miracles to explain why marsupials have similar genomes to each other and not to the analogs that we find of rodents that are placentals or moles that are placentals or carnivores that are placentals.
Miracles, yes.
These are all miracles. Oh, so they're all similar to each other.
Chris, I'm sorry to...
Okay.
I have a meeting at 9:30. We can commute. It's 9:30
right now. I can let you go.
I should have, out of courtesy to you, let you know I may have something if I can just check to see if they want me right now.
It's all right. I mean, we can end. It's we've got an hour and a half, and that's quite some time.
You're actually like 40 minutes. I did 40 minutes.
Yes,
we actually hit our target. I did want to say, I think evolution is theology pretending to be science. Okay. That was my point. This was all about theology. Evolutionism is not science. It's faithfully pretending to be science. There are books. There's a book that said Darwinism as religion. That's the point I was making.
All right. Good luck.
Yeah, so you can see Sal has no explanation whatsoever. If we return back to what Chris Bolt had to say, there's another clip, him being a little bit more explicit about this argument and trying to explain away the similarities that we see in genetic sequences, kind of spewing a bunch of word salad. So here's that clip.
So in terms of creation, we have common functionality in our human DNA, and it does function similarly to other created beings because they are in fact created just like we are. And yet human beings are unique. There is, yes, a common blueprint that's behind these things that comes from the archetype, comes from the mind of God himself. When we look at different processes in nature, be they physical or chemical or biological, we see that these things happen frequently, that it's not just all happenstance. We see that these things are reproducible, and we see that there are independent features from them in the organisms around us, different organisms in their genomes. But all of this just goes to show that we have a creator. The reason that there are differences in these genomes is because they are deliberately designed to do something different. We have different functions.
It's actually kind of hard to understand what his major point is there. I think what he's trying to say is that differences in genetic sequences between the different animals is due to different functions. Okay, sure, perhaps. The thing is, the KV1.1 gene essentially acts exactly the same way. Perhaps there's some very minor, subtle differences, but it essentially acts the same way across all mammals. There's no reason why the protein sequence of the KV1.1 gene in the gray short-tailed opossum should be more similar to the marsupials than it is to the rodents, even though the short-tailed opossum looks essentially like a rodent and it lives the lifestyle of a rodent. Yet it actually has much more in similar, the genetic sequence is much more similar to animals that don't look anything like it and clearly have very different lifestyles and completely different diets. Evolution offers an explanation for this, as you saw from Sal Cordova, someone who has been studying this issue for 30 plus years. His explanation for this pattern is miracle. It's not science, it's theology. It makes sense that Chris Bolt, a theologian, is the one spouting this nonsense, but it's nonsense, that's for sure. So, I've got a couple more videos that I'm going to be making about some of the things that Chris Bolt had to say. They won't be quite as long as this one. I mean, who knows? Maybe they will be. I tend to go on and on and on once I get started. But if you do like this content, please like and subscribe, and I will talk to you later. Bye.